Okay. A very good morning to all of you. I am Mr. Balasek Kale. Extend you a very warm welcome on behalf of Shri Venkatesh Arts, Commerce and Science College, Deulgaon, who has clubbed all of us for the international webinar on new horizons in chemical research and product development, organized by Department of Chemistry in collaboration with IQAC. Sri Venkatesh College is run by Sri Balaji Sansthan Deulgaon, Dist, Buldana. I am delighted to let you know that due to the continuous guidance and inspiration of hereditary trustee and president Honorable Raja Vijay Singh Jadhav sir, college has made remarkable progress in the field of education only because of his continuous efforts to uplift the standard of the college. Due to the outbreak of COVID-19 pandemic, education sector is also affected, and taking into consideration the need of the time, Department of Chemistry has organized this international level webinar. So on this occasion, I welcome Honorable Dr. S.D. Chavan sir as the president of this webinar, and I request sir to please join as a webinar. Yeah, to please join as a president of this webinar. Thank you, sir. I welcome the resource person of first session, Honorable Dr. Deulgaon sir, scientist first, Department of Radiology, University of Alabama, Birmingham, USA, and request sir to please join this international webinar as a resource person. Thank you, sir.
Thank you. Can I share my screen?
Okay, sir. Thank you, sir. I also welcome the resource person of second session, Honorable Dr. Nagarav Dhatrak sir, Associate Professor, Department of Chemistry, Savitribai Phule University, Pune, and request sir to please join this international webinar. I welcome the resource person of third session, Dr. Verla Andrew Virnapar sir, Department of Chemistry, IMO State University, Owerri, Nigeria, and request sir to please join this webinar as a resource person. Thank you, sir. I also welcome the resource person of fourth session, Dr. Nagnath Bhai More sir, Marie Curie postdoctoral researcher, University of Birmingham, UK. Thank you, sir. Thank you once again. Again, welcome. Sir, तुमची screen sharing थोडीशी stop. Thank you, sir. Again, welcome all the delegates, dignitaries, and participants who have joined this webinar. Thank you once again. So, to proceed to this webinar, I would like to request Mr. P.M. Kadam sir to deliver an introductory speech of this national webinar and introduce the resource person of the first session, Honorable Dr. Deulgaon sir.
Thank you, sir. So good morning to all the participants in this international webinar. Honorable Raja Vijay Singh Jadhav, Hereditary Trustee, Sri Balaji Sansthan, and President of Sri Venkatesh College, Deulgaon. Honorable Dr. Sudhir Chavan, sir, IQAC Coordinator and the President of today's webinar. All the resource persons, distinguished colleagues, delegates, professors, researchers, and students. I am pleased to welcome you to this international webinar on New Horizons in Chemical Research and Product Development, organized by Department of Chemistry in collaboration with IQAC. The Department of Chemistry of Sri Venkatesh College organized various curricular and co-curricular and extracurricular activities regularly for the students. So department has organized a national level conference in 2017, Frontiers in Chemical Science. It's also organized a state level PowerPoint presentation competition for the students in 2017. Departments regularly organizes the guest lectures, seminars, group discussions, quiz competition, and poster competitions for the students. So department has also organized some social awareness programs like harmful effects of fertilizers on human health, harmful effects of alcoholism on human health in the society. So, friends, we know as we are now going through the this COVID-19 pandemic situation, we all know the research in the science is helping us to recover from this situation. So, lot of research is done in the science and also in the chemistry, and it's still continue. So, the aim of this webinar is to enhance the knowledge about the research in chemical science and the latest advancement in the field across the world. So, the invited speakers of this webinar are internationally recognized experts in their discipline of research. So I am glad to announce that the webinar has got a very overwhelming response and about 900 participants have registered for this webinar. And the participants are from the various countries like Philippines, Myanmar, South Korea, Malaysia, Bangladesh, Algeria, Pakistan, Kuwait, Libya, Nepal, Oman, Iraq, Greece, etc. So the participants are from all over the world, and I hope this webinar will be a platform to gather and disseminate the latest knowledge in the chemical research. So I once again welcome to all the participants all over the world in this webinar from the organization committee. Now, I would like to introduce our first speaker of today's webinar. So it's a great pleasure for me to introduce the today's speaker, So who is going to talk us about synthesis of F18 labeled amino acid PET tracers for oncologic imaging. Friends, our speaker has made a powerful contribution to the field of research. He is working as a scientist one Department of Radiology, UAB School of Medicine, the University of Alabama at Birmingham, USA. He completed his MSc with university merit from Dr. Babasaheb Ambedkar Marathwada University, Aurangabad. He completed his PhD from National Chemical Laboratory, Pune in 2014. He completed his postdoctoral fellowship in 2018 from the Department of Chemistry and Radiology, University of Alabama, Birmingham, USA. Friends, he was awarded with Junior Research Fellowship and Senior Research Fellowship by CSIR, New Delhi. He also received a postdoc Research Day Award for oral presentation in 2018. He has been invited as a reviewer for more than 25 research publications in different international journals like Tetrahedron, Synlate, Organic, Biomolecular Chemistry, Synthetic Communications, and many more international reputed journals. So our speaker has a distinguished career in the research. So he has published many research articles in the international journals like Tetrahedron, Organic Biomolecules, Organic Letters, American Chemical Society, and many more. So I can't take the name of all the research journals. So he has a very good publication in the reputed journals. So he has also presented a paper in the various national and international conferences. He is a member of Society of Nuclear Medicine and Molecular Imaging, and he is also a member of American Chemical Society. So, friends, please welcome our first speaker of today's international webinar, Dr. Dattatray Deulgaon.
Thank you, sir. Let's move towards the technical session of this webinar to know more about the advancements in the chemical science and in the chemical research and product development. I would like to request Dr. Dattatray Deulgaon sir to share some knowledge in the field of chemical science.
Can you see my screen?
Yeah.
Yes.
Yeah. Thank you, Professor Kadam and Professor Kale. I'm Dattatreya. I'm working at University of Alabama at Birmingham in radiology department. And my research work is mainly focused on synthesis of different radio label PET tracers for cancer imaging. And I'm more focused at imaging of brain tumors. So I would like to discuss that research today with you all. So before I start my actual research, I want to let you know where from I'm exactly. So our university is located at Birmingham, state of Alabama, United States. We are here, you can see on this Google map, and this is our university campus. And my lab is right here around this side. So my lab is here around this area. So before I start my actual research presentation, I will like to give you a brief idea about what is PET. PET is positron emission tomography. This is imaging technique that uses radioactive substance to visualize and measure the metabolic and biochemical process in body. This is the non-invasive process, means in this process, we don't need to take any part of tissue or tumor from the body. And it provides the metabolical and molecular information about tumor biology. So this is very great compared to what we get information from MRI or CT scan, where we don't understand exact biology of tumor. So this is the example of PET tracer, CFDG, which is very commonly used PET tracer. And actually, this is the glucose analog. You can see here it is labeled with this F18 isotope, which emit positrons. So positrons are antimatters. They are opposite to electron, E+. So what happened once these drugs injected into human body, these positrons this isotope keep emitting positrons, and they collides with adjacent electron. As soon as they collide with adjacent electron, they form two gamma rays, and they are equal and opposite direction. So this detector detect the coincidence of two gamma rays. So based on that, this computer generate the image, and we can track where this drug is going exactly inside bodies. So this is how exactly these PET scanners are working. So at UAB, we have different PET scanners for imaging small animals for human clinical trials or human use. So this is here you can see this is small animal PET CT. Here we can scan mice or rat, those kind of things. And we have this PET MRI and PET CT for human scanning. And we also have this cyclotron facility available at UAB where we can produce different kind of isotopes. So I get my F18 isotope from this cyclotron facility for radio labeling reaction. And we also have other instruments like QTOF, NMR, different HPLC and radiosynthesis module here. So as I mentioned beginning, my work is more focused on detection of brain tumors. So this neuroimaging is very important in so this brain tumor imaging is very important in brain tumor patient because, hello? Is there any question? Can you hear me?
Yes, sir. The another guest has joined, so by mistake they have unmuted themselves.
That's fine. Okay, so I will continue. So this neuroimaging is very important in brain tumor patient because we have to first diagnose this patient and then plan the surgery and then monitor the radiotherapy response. So for everything during this neuroimaging is very important. Right now we have two methods are available. One is the MRI, contrast enhanced magnetic resonance imaging, and other is PET agents. So they are, EFDG is very commonly used PET tracers. So, but the problem with these both techniques, they have some kind of limitation. If you talk about MRI, it has limitation to detect non-enhancing part of tumor. So what is non-enhancing part of tumor? It is the part of tumor where BBB is not broken. BBB, blood-brain barrier. So if in this MRI, they use this gadolinium ion as a contrast agent, and they do not cross BBB. So if you have some part of tumor where BBB is not broken, this reagent cannot go inside, and so we cannot see that part of tumor. Even MRI has problem, we cannot detect whether that tumor is really tumor or this is some abnormality after treatment or something, or recurrent tumor from treatment effect. So these are some limitation of MRI when we talk about brain tumor imaging. PET tracers are very good for different kind of cancer imaging, but for when we come to brain tumor imaging, even this FDG is also has limitation. As I mentioned, this FDG is glucose analog. So what happened? So what happened with this glucose analog, brain always take a lot of glucose, brain needs a lot of glucose. So, and if you have brain tumor, so this glucose can go equal amount in normal brain and in tumor tissue. So it is very difficult to distinguish between normal tumor, normal brain and tumor with EFDG. So EFDG cannot be really useful in brain tumor imaging. There is another tracer which is available, which called FET, which is tyrosine derivative. This is good for brain tumor imaging, but the problem with this FET, it has very low contrast imaging. It transport into cell via system L transporter, and which has reversible uptake. So it get washed out from cell very quickly. And right now this FET is only available clinically in Europe. In USA, it is still under useful only for the research kind of purpose. It is not for like daily clinical use available. So this is example that I want to show you what is exact enhancing and non-enhancing region. So this is patient with glioblastoma, brain tumor, and you can see this area under this yellow line, it has tumor here. And with this T2 pre means before injecting contrast, you cannot see anything. But after contrast, you can see this some part of tumor is getting enhanced. This is white part. So this is the part of tumor exactly where BBB is broken and where the gadolinium contrast agent can cross. And so we can see this part of tumor. But the problem is here, the tumor is bigger than actually what we see in this enhancing region. Even with T2 experiment, this all area is getting enhanced, but this is not all the part of this enhanced region is really a tumor, because there is some abnormality also. So this is the limitation with MRI. So we cannot really see the what is exact boundary of tumor, non-enhancing part of tumor. So that is why it is very important. We should have the new techniques or PET tracers that will give you idea about a whole tumor, not only non-enhancing part, enhancing and non-enhancing part both. So aim of our lab to develop novel radiohalogenated amino acid PET tracers, more focused on neuroimaging. And we generally make amino acid based PET tracers. Why amino acids? Because what happens in cancer cell, there is alterations in cellular metabolism. Cancer cells are very aggressive, so they need a lot of amino acids. So what we do, we synthesize different radio-label amino acids, so they get into the cancer cell in more amount compared to the normal cells using different amino acid transporter. And based on that, we can detect where exactly cancer is located inside body. So right now in my lab, we are working in these 4 different amino acid transporter. This first one is system L, which generally transport large neutral amino acid like leucine and tyrosine. And we have this drug FET for this kind of purpose. And this has reversible uptake. So it get washed out from cell after some time. And this is another system A, which I talk more in detail. This generally transport small neutral amino acids like alanine and serine. And we have these drugs, Methyl FMP. This is developed by Dr. McConaughey. And this is another example, methyl AIB, which is carbon 11 derivative. And this has concentrative mode of action. So these kind of drugs get accumulated inside cells and we get very high contrast images. And this is another system which is called cationic transporter, which transport positively charged or basic amino acids like histidine and lysine. And this drug AFATP is also developed in our lab for this system. And we also work on this other system A ASC, which is called allele, serine, and cytosine. So this is just short forms which transport neutral amino acids, and we have this FACBC drug for this molecule, this transporter. And one thing I would like to tell you here, that systemial and cationic amino acid transporter, they both present on BBB, blood-brain barrier, luminal side of BBB. So if you have any drugs between system L and system A transporter, they can cross BBB, but system A and system ASC, they are not present on BBB. So there is limitation with these two transporters. So I'm going to talk about more this drug Methyl FMP that we work in our lab. The story of development of Methyl FMP started with this methyl AIB, which is also very good for brain tumor imaging. But the problem with this methyl AIB, it is C11 derivative, and this carbon 11 isotope has half-life only 20 minutes. So if you make some drug, every after 20 minutes it gets half and half, so it is very challenging to work with this kind of drugs. So if you want to make it synthesis same day and you have to scan the patient, this is kind of difficult. So we developed this methyl FMP tracers. This is F18 analog. And so because with F18, it has half-life around 110 minutes, almost 2 hours. So these drugs can be used for longer time. So as if we make F18 tracers, so that generally have high metabolic stability because these CEF bonds are kind of stable, and they can easily, this F18 can be easily replaced by hydrogen, oxygen, halogen groups in drug molecules. And because of longer half-life, it is possible to distribute these PET drugs to adjacent PET CT facility for human scanning. And one more advantage that, that we can scan patients for late time points. What happened, it is not like as soon as we inject drug molecule into patient that it will go to tumor and we will get good image. Sometimes we have to wait like 4 hours. So with this carbon 11 derivative, it is not possible to wait that long because it has very short half-life. But with F18, we can even scan up to 4 hours late time points. So that is advantage with this F18 PET tracers. So this methyl FMP, it has very high uptake in glioblastoma, as well as other cancers like prostate cancer, breast cancer, lung cancer, and ovarian cancer. So this is very versatile PET agent. And this is another example where I want to show you why we prefer methyl FMP over the available PET tracers. So this images from intracranial mice model, which has a brain tumor here. So this part, dark part, is where the brain tumor is. And this is the brain part of that mice. So this first 3 images are PET, and this is PET and CT fusion. So you can see with FDG, there is not really clear distinguish between tumor part and the normal brain part. So it has very poor tumor to brain ratio. It is almost 1:1. With FET, we have better ratio. We can see the tumor better here, but still the ratio between tumor to normal brain is 2:1. So still the boundary of tumors are not very clear. So as I mentioned previously, it has reversible uptake into the cells, so it get washouts, and so we don't get very clear boundaries. And with methyl FMP, you can see here, you can clearly see what are the boundaries of tumor, and the ratio for tumor to normal brain is 9:1. So it is really better compared to FET and FTG. And this is another image where you can see this is again the mice model. The down is model with radiation necrosis, and the top model is DBT tumor model. So you can see in this tumor model, this is Yamara, and these 2 are PET images. So you can see the methyl FMP stays inside tumor even after 4 hours. But if you see in this radiation necrosis, it get washed out after 4 hours. So what is radiation necrosis? Radiation necrosis is when after radiation therapy, there is some kind of injury and those are there into brain. So that is called, we call radiation necrosis. So we are planning to do first in human study with methyl FMP at UAB. We hope by early next year, before summer, we can scan our first human patient with methyl FMP. And our initial focus for this study is distinguishing between radiation necrosis and recurrent tumors or metastasis. That is our initial aim to scan for these patients. And so this is the synthesis for methyl FMP precursor. We want to use this tracers for humans. So FDA is very strict about purification, how we make that tracers, everything. We have to keep the record for everything. And we want to make sure everything is pure. There is no any kind of contamination. So we start this synthesis with this alpha methylserine, which is commercially available. And then I do protect this OH group and acid group, and also this NH2 group. So NH2 group I protect with BOC, and this acid group with tertiary butyl group. And this is not very complicated synthesis. Synthesis is straightforward. There is no chance of racemization here. And after that, after I protect this both acid and amine group, I treated with this thionyl chloride pyridine, get this cyclic intermediate. And after oxidation with ruthenium chloride, NaI4, I get this cyclic sulfamidate ring, which is key intermediate here. And then I replace this BOC group and put here a N-methyl group here. So this is our actual precursor for the radio labeling of methyl FMP. So I will talk more detail about how we do exactly this radio labeling reaction. So this is just all spectras, NMR, HPLC, mass resolution, high mass resolution, elemental analysis. So what are the main requirements of radiosynthesis reaction? So to do any kind of radiosynthesis, the reaction and purification should be done in short time, because I mentioned this kind of radioisotopes have very short half-lives. So we want to make this reaction quickly purify them. And this reaction should be reliable, efficient, and inexpensive. And generally in this kind of reaction, precursors are present in large amounts. So we have to keep that in consideration. And we should have the high molar activity at the end of synthesis. Molar activity means activity per mole. Activity, I'm talking in the sense of radioactivity. And we also want to make sure that whatever reaction vessels or equipments we use for this kind of reaction, they are properly shielded, because we don't want to expose ourselves with radiation. So generally we do all this reaction behind lead chambers. So lead generally absorb gamma rays, so we don't get exposed to those radiation. And obviously, final product should be injectable form to do any kind of preclinical or clinical study. So this kind of things we should need to take in mind when we do the radiolabeling reaction. So what happened, I get this F18 from this cyclotron facility. We have this TR24 cyclotron. What they do, they take this O18 water. O18 is a stable isotope, and they bombard that one with proton. So this proton displays this neutron from O18, and we get this F18 isotope. So this is how they produce F18, and this is how I get F18 from cyclotron. This is in O18 water, it has F18 ions. So once I get this F18 from cyclotron facility, then that I use next that for radiolabeling reaction. And this is the hot cell that or lead chambers that I'm talking. So we don't want to get exposed to ourselves. So we do all reaction behind this lead chamber. So even this glass is also made of lead, and everything this is made of lead. So, and these are manipulators. So we don't need to directly touch this vessels here. So you can just handle this manipulator from outside. Similar action will take place inside. Or we can also use this automated radio module. So here they have different kind of vials. So before you start reaction, you can add all your material, and you can just control this all module using computer from outside. It has different reactors here, vials. You can transfer from one reactor to another reactor, your reaction mixture. I generally do 3 steps reaction inside this module. It has HPLC also. So this methyl FMP synthesis I develop on this module. And once we make program, you don't need to every time do that reaction. You just start that program, you add those reagents, reaction will go automatically. Only we have to be careful when product is coming out from HPLC. So once I get EF18 from cyclotron, I capture that on QMA. So what is this QMA? QMA is quaternary ammonium salt here, which capture EF18 ions and separate O18 water. So we remove that excess O18 water. And then once this get captured on QMA, I pass with this cryptofix and K2CO3 solution. So this base replace this F18 ions and we get this EF18 and cryptofix inside our reaction chamber. So this cryptofix crown ether, we just use to make it F18 more nucleophilic in our reaction. So this is what reaction goes. So once I trap F18 into my reaction vial, then I add 1 mg precursor. So this is what you can note here. So we need very, very less amount of precursor for synthesis of PET tracers. I just use 1 mg precursor for this reaction. And then I heat this reaction at 85 degrees Celsius in acetonitrile for 20 minutes, and then it undergo nucleophilic opening of this cyclic sulfamidate ring. And we get this radio label compound as well as this impurity. As I mentioned, even this is 1 mg, it is still so much precursor for 3 curie. So I get always this impurity because of some moisture into reaction or something base is there. So we get this hydroxy impurity. And once this radio labeling happen, I do hydrolysis to remove this N sulfur bond. And then I do final purification on HPLC column. So before I go to column, I use this Dynex cartridge, which neutralize excess acid because this column is sensitive to acid. And there is another cartridge I use here, HLB, which remove any other organic impurity, which is non-water soluble. This is also organic, but it is water soluble salt. So it don't get captured on the HLB. And then I purify and I get this final product labeled Methyl FMP. Generally, if I start with 3 curie product, starting material, I get 750 millicurie product after 90 minutes. So total this reaction takes 90 minutes. So all this reaction takes place on this module and we can control with computer from outside. So this is just simplified diagram for this module. So here I do trapping of F18, and once F18 trap, I do the radio labeling and hydrolysis reaction in this 1-part reaction. And then after neutralize, I transfer into this second reactor. From here, it undergo, it get injected into HPLC, and after HPLC, we get collected product here in this vial. This is the final collection. It goes through this V27 to then this 4th vial and then into the product vial. So what is advantages of automated radiosynthesis? We can do this synthesis manually also, but if we do this synthesis with automated module, it can be efficient and fast because this has very short half-life isotope. So we need to make it very fast, and we can avoid the manual errors, and it will have reproducibility in terms of purity and yield. So these are the advantage of radiosynthesis, automated module of radiosynthesis. And with this kind of synthesis can be used in GMP facility for the routine production of these drugs for human use. So this is just a HPLC chromatogram. I just want to show you the purity of this compound. So obviously we don't make any other isomer, but we just want to make sure that we are making correct R isomer here, which is useful. Yes, isomer is not very useful for us. And we just use this Aztec column. These both are actually this is non-UV active compound, but if we use this Aztec column with copper ions into mobile phase, it forms some complex with acids and amine, and we can see this kind of non-UV compound amino acids with wavelength 254 nanometer like that. So in summary, we make this methyl FMP production with 50% of decay corrected yield. Means if I start with 3 curie, I get final product 750 millicurie after 90 minutes. and molar activity, like concentration, is more than 1 curie per micromole at the end of synthesis. And for one patient, we just need to inject 5 millicurie to 10 millicurie dose. Means one patient, we just inject less than 1 microgram actual drugs. So we are injecting very, very less amount of drug in one patient. So there is not much worry about toxicity here. And radiochemical yield and enantiomeric purity for this synthesis about 99%. And the drug, what we make here, the pH for this drug is around 4.5 to 5%. And this is the isotonic solution, which is important if you want to make it injectable into the patient or any animals. So right now, we are doing our best to, as soon as get these drugs into humans. We are about to submit our CMC to FDA. We hope we will get very soon approval from FDA. And our initial focus, as I mentioned before, we want to differentiate between radiation necrosis and gliomas. And we are also working on other application of methyl FMP, not only in just gliomas, but also in renal cancer. And also we are trying to develop new immunoviral therapy with methyl FMP, also developing new glucose inhibitor drugs. And also we are working different other PET tracers also in our lab. So with this one, I would like to stop. I would like to thank Dr. Jonathan McConty, Director of Division of Molecular Imaging and Therapeutics. Dr. Susan Lappy, she's the Director of Cyclotron Facility. Dr. Norio Yasui, he's my lab mate, cyclotron team member, UAB. And we get research funding for this project from UAB and NIH. And I would like to thank all of you, all the member of Shri Vyankatesh College, Deulgaon Raja, for invitation. This is really great. Right now, social distancing is important, and this is a really great webinar. And I wish you a happy new year to all of you. Hope in next year, everyone will get their vaccine very soon. If you have any question, please feel free to ask me. I will try my best to answer. Even you can reach me through my email, d.devrankar@uab.edu. Thank you.
Sir, thank you, sir. Yeah.
Hello.
Yes, sir. I want to ask one question.
Yeah.
Sir, is there are some side effects of these tracers?
Not tracers, it's radioactive, and what activity we inject, like that is 5 millicurie. And if you see what is the advantage of these drugs compared to that, whatever the disadvantage with that side effect of that much radioactive inside, radioactivity inside body is negligible. Means generally nothing happened, but we can ignore those kind of small side effects compared to what are the advantage of these methods.
Oh, okay, sir. Thank you.
And it's depend on drug to drug. And generally, as I mentioned, we inject very small amount of drugs into patient. So we never really reach to that toxicity level for that drug.
Okay. Any more question from participants?
There is one question that is not related this topic, but I will just ask how to separate the drug by HPLC technique from plant.
Sorry, I didn't understand. How to separate drugs?
By HPLC technique.
HPLC, yeah, HPLC, that mobile, that stationary phase has different retention with side products and your actual drugs. So they come at different time points. So my drug comes at from 19 minutes to 22 minutes, and side products comes later, after 2-3 minutes later. So we can collect initial product when we see drugs is coming, and we can stop collection when side product is coming. So it's easy to separate them. It's really depend on what is the retention time of your drug and your side products. Is that I answer your question? Hello.
Thank you, sir.
Yeah.
Thank you very much, sir.
Okay.
For your valuable information. Sir has explained amino acid transporters and tracers in very simple and easy way, and also sir has put some advantages of F15 labeled PET tracers before us. So thank you for this informative talk. So now I request Dr. D.R. Birade sir to introduce the resource person of second session, Dr. Nagar sir.
Okay, good morning, everyone. My name is Dr. Dikshaputra Ramprao Birade, and I am happy to introduce today's guest speaker, Honorable Dr. Nagaro R. Dhatrak sir. Dr. Dhatrak sir is working as an associate professor in the Department of Chemistry, Savitribai Phule, Pune University, Pune. Dhatrak sir completed his undergraduate and postgraduate education from Amravati University, Amravati. He qualified SET exam in 2003 and NET exam in 2004 and awarded PhD degree in 2016 from Savitribai Phule Pune University, Pune. He has more than 15 years of teaching experience and research experience. His area of research is organic synthesis and electrochemistry. He is also interested and working on developing instruments which are used for chemists. Dr. Dhatrak sir is published number of papers in national and international journals as like Tetrahedron Letter, Tetrahedron Asymmetry, European Journal of Organic Chemistry, and ChemCom. He has completed 5 projects under the funded by UGC and BCD and other agencies. Along with this, he has written 2 books and chapters for a number of books. Now we got an excellent opportunity to listen something more interesting from such a great personality. He is going to share his research experience on total synthesis of canonidine alkaloids. So I heartily welcome Dr. Dhatrak sir.
Hello. Hello, organizers. Please confirm from your side that everything is all right and you are. I'm audible.
Yes, sir. You are.
Yeah. Yeah. Okay. Thank you. Thank you very much. So once again, please confirm that this slide is visible to you. Excuse me.
Sir, now try to share the screen.
Yeah. Okay. Yeah. Is it visible now?
Now, yes, it is now visible.
Yeah. Yeah. Okay. 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 So I thank organizers for giving this opportunity to me. And I'm going to talk on a total synthesis that actually has been carried out in my laboratory, and recently we are working on it. But this is one of the interesting total synthesis that we are actually taking interest in it, and only 2 to 3 research publications are available on that. But humanity needs this alkaloid to a greater extent, and therefore we are working on it. Okay. So actually, I'm going to talk about the total synthesis of this alkaloid. This is very typical alkaloid whose name is the canonodine. Okay, so it is actually a pyridine alkaloid, is having a 7-member ring fused with it and a tertiary alcohol group here and one methyl group, isn't it? And there are 2 stereocenters, and these 2 stereocenters are essential for the pharmacological activities of this molecule. And we are trying to synthesize this molecule in the laboratory. The reason is this molecule is naturally occurring and it is extracted from the leaves and the fruits of the ylang ylang plant. So that plant is also called as Canaga odorata plant, isn't it? And very trace amount of the alkaloid is actually present in the plant, and it is not possible to gather the required quantity of the alkaloid from the plants only. And even if we try to do that, it will damage the nature, isn't it? And therefore, we are trying to fabricate a method, easy and the efficient method for this total synthesis of this molecule called canonodine. Okay, so this is what the background of the research, isn't it? So the Canaga odorata, it is a sort of the plant, tropical plant, subtropical plant, angiosperm. It's flowers actually, and its oil is being sold on the many websites, and it's readily available. And the oil is also having a small quantity of the alkaloid, isn't it? But that is not sufficient to execute any pharmacological activity, isn't it? And therefore, there is special need to synthesize this alkaloid in the laboratory, and our efforts are there. So these are actually the flowers and the fruits of that particular plant, isn't it? And the alkaloid is separable from these flowers as well as fruit. Even it is discovered that the alkaloid is spread throughout the plant, means irrespective of the fruits and the flower, but more concentration was discovered in the fruits and the flowers. Okay. So what is the utility of this alkaloid? The utility of this alkaloid is that, so it is selectively toxic to the hepatocellular carcinoma. Okay, so hepatocellular carcinoma. What is the hepatocellular carcinoma? In simple language, it is a sort of human liver. It's sort of human liver. And this is a very strange situation that for the human cancer or human liver cancer, there is no medicine present till the date, isn't it? And therefore, there is a special need. Actually, this medicine is being used in the folk medicines, means this alkaloid or this plant, Canaga odorata, this plant is being used in the folk medicine to cure the many diseases, and it has selectively discovered that this is having a selective toxicity to the carcinoma cell. What is the selective toxicity? Selective toxicity, it can only kill the human liver cancer cells, and other cells remain unaffected. So that is the great thing with this drug molecule, or with this alkaloid. Okay. So this is one of the publication which has published in the JOC in 2006, and they have synthesized this molecule using a large synthesis. Actually, it involves a total 29 steps, and this synthesis very much cumbersome synthesis, but it's a great achievement. That is, finally they could get the molecule in hand, isn't it? So this is one initial synthesis on the molecule. Okay. So where to start? We thought that this molecule, the final molecule, is having 2 stereocenters. Okay. So at least start with one stereocenter from the starting material. Okay. So we applied the chiron approach, isn't it? So it is clearly shown over there that another, that is the methyl stereocenter, that this methyl center, methyl group can be synthesized later, but one center we are actually carrying on, isn't it? Okay. One more thing that was hurdle in the total synthesis was that we started from the carbon. The carbon was the principal requirement for the synthesis. But the problem is, the S carbon is 10 times more expensive than the R carbon, isn't it? So we decided to complete our synthesis from the R carbon, that is the, and the second synthesis we can do with the S carbon, because R and S are chemically same reactive. So whatever synthesis we would carry out with the help of R, that can be extrapolated to the S isomer, isn't it? So S will actually give the original molecule, but the R will give its epimer. So we are currently engaged in the synthesis of the epimeric form of the canonine. Okay. So this is what the retrosynthesis analysis was planned, isn't it? Okay. So this is the actual molecule we wanted to synthesize. So it is having R and R stereocenters. A tertiary alcohol is there, isn't it? So tertiary alcohols are very, very reactive and it's difficult to handle them. So with the help of FGI, we started working on that, this work. So we converted, means we thought that it will be proper to work on isopropenyl group than the tertiary alcohol. Okay, so where to get from this isopropenyl group? You see the carbon also is having the same isopropenyl group with the same stereochemistry, isn't it? Okay. The rest thing is that the pyridine ring can be synthesized with the help of one diketone synthesis. So we need a 1,5-diketone moiety so that we can install a pyridine ring into it. Okay. And how to get this? So this diketone can be obtained from this molecule, means a keto ester molecule. And the keto ester molecule in turn can be synthesized by this one, this molecule. This is nothing but a cyclopropanyl type of the system, bicyclic system. And this bicyclic system can be synthesized from the ES carbon, isn't it? Okay, but ES carbon is very, very expensive. So we started our synthesis from the R carbon. Okay, yeah. So this was the proposed scheme, but many times we think that this scheme would process, but there are many, many hurdles in the actual synthesis. Okay, so we started the synthesis from this particular elide, salt elide, isn't it? With the help of the NH that can be converted into the elide. And we thought that this elide will react with the carbon and we will get this cyclopropyl system, isn't it? And this salt can be easily synthesized back from the DMSO and the ethyl bromoacetate, isn't it? But actually we synthesize the salt, we synthesize the elide, but this elide refrained to react with the carbon. We could not get this molecule. So this particular scheme was having a setback at the initial steps only. So we discarded this particular scheme. Okay, we went ahead with the another retrosynthetic scheme. Okay, so in which we actually thought that we should get this particular molecule, isn't it? It is nothing but a heptanone system, 7-member ring with a 1 stereo center. There is facility for the another stereo center because there is a carbon-carbon double bond. So a prochiral carbon is already exists in the molecule, isn't it? And we started, we can start the synthesis with this one, isn't it? This also arrives at a 1,5-diketone, so that with this, with the help of pictet GAMS modification in the Hans pyridine synthesis, we can install the pyridine ring into it. Okay, so in this way, this was the retrosynthetic analysis part 2. Okay. Okay. So as the carbon S is 10 times costlier than the carbon R, okay, we thought that we should start with the carbon R because when initial reactions are trained, a lot of starting material is required, isn't it? And it was indeed cost effective to work on the carbon R than the carbon S, isn't it? So the result of the carbon R, actual anticipated molecule is this one. But if we start the reaction with the carbon R, we will get this molecule, isn't it? So we started actual synthesis of this one. So the retrosynthesis analysis shown over here. So the tertiary, instead of handling the tertiary alcohol, we started with the isopropenyl one. This group, we thought that this group can be comfortably manipulated or handled. Then we envisaged this molecule, that is the 1,5-diketone system. So this molecule can be converted into the pyridine ring, required pyridine ring with the submethyl substituent with the help of Hans pyridine synthesis and a pictet GAMS modification. Okay. For this one, this molecule, we needed this one. So heptanone system with the required stereochemistry was anticipated there. Okay. And yes, this can be synthesized from this one. Okay, so selective reduction of the double bond or stereoselective reduction of the double bond would establish the R stereo center at this particular position that was anticipated or envisaged. And this molecule can be synthesized from this one. And how the synthesis goes ahead, that is given over here. Okay. Now, actually, we started the synthesis from the DMSO. So DMSO, upon reflux with the methyl iodide for 48 hours, we get the trimethyl sulfoxonium iodide salt. Okay, this is a very simple preparation, and we can get a large quantity of the trimethyl sulfoxynium iodide, which is otherwise very costlier in the market. Okay, so DMSO actually is heated under nitrogen atmosphere, and methyl iodide 1 equivalent is added to it. So after nearly about 48 hours, the entire flask become solid, and the crystals are gathered inside. Those crystals can be filtered with the help of a Buckner funnel under vacuum, and you get such type of the material, that is the black material. This black or the purple color is because of the iodine impurities that actually come along with the salt, isn't it? And that can be washed several times with the help of chloroform, and we get a brilliant white salt, and that is nothing but the trimethyl sulfoxynium iodide. And this material was actually required. Okay. So once the trimethyl sulfoxynium iodide was ready, that in a reflux assembly, along with the dry THF, trimethyl sulfoxynium iodide and NaH was taken. And the salt is converted into the corresponding sulfur halide. Okay. So how that reaction goes? So the trimethyl sulfoxynium iodide here, means NaH when operated, one of the acidic hydrogen is removed, and see, this is the halide we require. This halide is generated in situ. So when the halide is generated, a carbon was added into the system. One equivalent of the carbon is added to the system. Okay, and it actually brings about a Core-Tchaikovsky reaction, and we get such type of the system. So this is a bicyclic system. Double bond is actually cyclopropionated, and we know the cyclopropyl rings are strained ring, and they have tendency to open open easily, isn't it? And this is the reason because of the 6-member rings opens up into the 7-member ring. So the main intention behind synthesis of a 7-member ring was sufficed over here. So the carbon was cyclopropionated with the help of the Core-Tchaikovsky reaction, and we get this particular system. We could not isolate this, neither there was need of isolation and purification. A small quantity actually was purified, and then it was used for the characterization purpose. That is, we are getting the anticipated molecule. Okay. So this molecule was then reduced with NaBH4 in aqueous MEOH, aqueous methanol, isn't it? So this particular ketone function is converted into the alcohol. So you see in the what we got in the product. In the product, we got the ketone function was converted into the alcohol. Okay, but simultaneously, we also got this particular one. So there is spontaneous ring opening, and we get a 7-member ring with one alcohol moiety into it, isn't it? So this is the ring expansion. Okay, but sorry, here it is not the major one, it is the minor one, and this is the major one. Okay, a small trace of the amount was generated, this one. So it was necessary to expand all the molecule, and for that an acidic medium was used. So the latter reaction was carried out in acetone. It went for 24 hours, and the catalyst or the reagent was 7% HClO4, okay, perchloric acid solution. So upon stirring in the perchloric acid, all the molecules were expanded, means ring expansion took place, and we got this particular cycloheptanol system at 65% of the yield. Okay. Later we thought that we should convert this alcohol function into the ketone function. So oxidation was necessary. So we carried out oxidation with the help of PCC in DCM at room temperature, and this oxidation goes well. But there is one problem. That problem is, rather than to get a single molecule, we actually get a mixture of 2 different molecules. So this is UV inactive, but this is UV active because there is alpha beta unsaturation, or the conjugation is there with double bond. This is the non-conjugated system, and therefore this is UV inactive. But we got both one. The migration of the double bond was because of the acidity of the PCC. However, this was the anticipated molecule, and this was obtained nearly about at 30%, and this was obtained at 70%. Okay, so this was in line of the synthesis, but it was necessary to convert this molecule into this molecule, as this was the anticipated molecule. Okay. So then this molecule was reacted with the butoxide in tertiary butanol, and this double bond was to migrate at alpha beta position. So we got a alpha beta unsaturated ketone function with 7-member ring and 1 stereo center. This was then fully analyzed with the help of a NMR, C13, mass IR, and mass IR, and even the polarimetry. Okay, means optical rotation was also noted. Okay, so once this molecule was necessary synthesized, there was a special need to reduce the carbon-carbon double bond. So this double bond was no longer needed in the synthesis. But one more thing is there, that whatever the double bond and the prochiral center or prochiral carbon is, that carbon was to reduce stereoselectivity, isn't it? So we applied a variety of this reagent and we tried to reduce the double bond. Okay. So with the help of LiH4, what happened? Generally, we know that LiH4 carries out 1,4 reduction, but instead of 1,4 reduction, we got these two molecules with the help of LiH4 reduction. So completely reduced molecule and partially reduced molecule. But this was the measure in which only ketone function was reduced and double bond was remaining as it is. So LiH4 was not a good choice. Later, next choice was a Zedian ACOH system. Okay, so that worked well, isn't it? So that was also worked well, but there was a thermal degradation of the product because zinc acetic acid reduction was thermally carried out. Okay. Zinc AOH, that was also not good. And this lithium EDA, this was work there, isn't it? So this is nothing but a sort of Birch reduction, or we also call it Benkesser type of the reduction. So that worked well. So zinc ACOH worked well. Then MgOH, Mg, that is magnesium in methanol, that also worked well, but that needed a oxidative process at the end. Okay, but the problem was that whatever the Gillespie's matrix generated in the course of the reaction, that was very difficult to handle, difficult to filter, and therefore we discarded this one. Okay, with the help of NaBH4 and TBOH, it did not work. Only carbonyl function undergoes reduction. And one more important asymmetry reduction we carried out was the reduction using the SECFOS and the copper hydride under diminished temperature. That worked well. So these were the probabilities and the possibilities tested for reduction of the carbon-carbon double bond, which was otherwise not required. Okay, so this is the one of the best reduction that was worked in our laboratory and converted the double bond into the single bond. The double bond was reduced. So that was the Birch reduction. It required liquefaction of the ammonia. Okay, first drying of the ammonia was necessary, and then liquefaction of the ammonia was also necessary, and the Birch reduction was carried out. Okay, so this was actually a very tedious process and dangerous also. Any elevation any elevation in the temperature of the reaction would lead to a catastrophic explosion of the reaction vessel. And therefore, we thought that this is not suitable in the laboratory, and we discarded it. We're working on the Benkesser's reduction was possible. It required just lithium metal, ethylene diamine, isopropyline, and 0 degrees centigrade. So you see here in Birch reduction minus or negative temperature is required in order to liquefy the ammonia. But in Benkesser reduction, the reaction can be carried out at 0 degrees centigrade, and reaction was too simple. This observation was later published in the Resonance. So you can see the Birch and Benkesser reduction. This is one of mine paper that elaborates the process of the Benkesser reduction. Okay, so with the help of Benkesser reduction, we could get this molecule. But this molecule was in distereomeric in nature. One stereo center was there, but we did not have any control over the generation of the second stereo center. Okay, and that was the problem with it. So because of this problem, we have to alter our synthesis. Okay, so this is one attempt at how we begin with the Benkesser reduction, and we got one more important molecule. So This alpha beta unsaturated ketone was converted into this type of the enolate, lithium enolate. And this lithium enolate was trapped in the ether and trapped by the MVK, methyl vinyl ketone, and we get this diketone. Surprisingly, this diketone was yielded in a trace yield, okay, but it was noted that this is also an important alkaloid, natural product, not alkaloid, important natural product called chabrolidine. So during this entire synthesis, we also got a one more natural product in the root, isn't it? It was in trace quantity. It is characterized fully, and a small quantity of is available nowadays. Okay. The most successful method for the reduction of the alpha beta double bond was NaBH4 and the pyridine, isn't it? At the diminished pH, only double bond was reduced, isn't it? And we get a mixture of the compounds, this alcohol as well as the ketone. The mixture was then oxidized, so we get this compound. But in this also, a diastereomeric and inseparable mixture of the diastereomer was generated, okay? And we did not have any control on the stereochemistry of the methyl group. That was the problem, okay? Anyhow, when this molecule was generated, it could give us this molecule. So we started working on this molecule. So with the help of LDA and the NTHF, we actually enolized this molecule. So lithium enolate was then trapped with the help of TMSCl. And this ceryl enol ether was reacted with this one in presence of ZnCl2 on Al2O3. Okay, so after just stirring, we got this particular molecule. So this was one more method for the synthesis of the chabrolidine. Okay, so this is the anticipated 1,5-diketone that was required for the establishment of the pyridine ring. Okay. Once this was done, we started the synthesis with the 1,5-dione that we synthesized. So this 1,5-dione upon reaction with hydroxylamine in ethanol under reflux condition, it undergoes a Hans pyridine synthesis. Actually, it is a peptide GAMS modification, and it gave us these particular molecules. So anyhow, we could synthesize these molecules. Okay, but there are certain limitations of this particular synthesis. What is the limitation? Instead of getting the pure molecule, we actually get the molecule in its epimeric form. So we did not have any control on the stereochemistry of the methyl group. How to obtain the control on the stereochemistry of the methyl group? So we started inventing into that. So we again went back to the alpha beta unsaturated ketone that was earlier synthesized. With the help of R-DTMB sec-phos, this is the asymmetric reagent, and copper hydride at diminished temperature, we could get a required stereochemistry of the methyl group, isn't it? It shows a optical rotation of -62. So this was a bigger achievement in the laboratory. So the structure of the DTMB sec-phos, this is actually expensive, but it requires in a catalytic amount, so reaction is cost effective therefore. Okay, it requires a copper hydride and then PHMS, and this reaction also work well, gives 60 to 65% of the yield. Okay, so a stereo pure form of the cyclohexanone system, required cyclohexanone system was ready with us. So we started this next synthesis with the help of this one. So again, this was enolized. So to get the lithium enolate, the lithium enolate was then trapped with the trimethyl and silyl enol ether was generated. And this silyl enol ether was then reacted with methyl vinyl ketone to get the diketone. And this diketone was converted into this molecule. This molecule is having a isopropenyl function. And this, we are thinking that this isopropenyl function can easily be converted into this one. But there are certain problems. Once in the attempt of the acidic hydrolysis, this won't work because this is actually destroying the stereo center, and we are therefore epoxidizing it and using some other method, and the next process is going on. Okay, so the best feature of this synthesis is that this is the natural product we are getting. This is called a chabrolidine, and this is the next natural product we are getting. Its name is bigopyridine. So in the one total synthesis, we are actually hitting the three different natural products, chabrolidine, as well as the epigopyridine, and the last one is the canonidine. So this is actually the total synthesis we actually carried out in the laboratory. So actually you see the whole total synthesis is sum up in a circular form, in a sequential form. So we actually started from a natural product, that is carbon, that is obtained from the mint leaves, and we are actually arriving on the total, arriving at a another natural product which is very important for the humanity, that is called canonidine. And in the synthesis, we are getting several natural products, like this is chabrolidine, this is one more natural product, this is the epigopyridine, and this is the canonidine. So we are trying to hit up all these things. Our synthesis is at this particular stage, and we are just one step away from the total synthesis. But meanwhile, as the this corona pandemic emerged, and we have to stop our synthesis. Okay, so we are at this particular position. Okay, so this is how the synthesis went around, and we have converted one particular natural product into the another natural product. So this is nothing but a very important total synthesis. Okay, so what are the other natural products we are getting in this synthesis? So at step 8, we are getting this chabrolidine. Okay, and this is actually obtained from soft coral, sea coral. So another one is this natural product, that is epigopyridine. So this is obtained from the Pogostemon patchouli. Okay, so this is one more benefit of the synthesis. And at step 10, we are envisaging that we will get this particular natural product, main target product, that is called canonidine. Okay. So this is the other natural product based on the 7-member scaffold. We are also trying to convert that 7-membered cycloheptanone system into the acetylene. So this is one more project going on. Then this S plus acetylene that can be synthesized with the help of the same method. Okay, and the 1,5-diketone method for the acetylene synthesis is given over here. That the diketone can be simply converted into the by Wittig reaction, that can be converted into the this dyne system. Okay, and the dyne system then can be subjected to Gruff's catalysis, and the acetylene will be ready, isn't it? So this is one more side synthesis going on in the laboratory. Okay, so I thank the Vyankatesh College for giving me this opportunity. And I also thank the colleagues from the Vyankatesh College for organizing such a nice seminar. So thank you very much. In case of any questions, you can revert back to me.
Thank you, sir.
Yes.
Is there any question from participants? All the participants are requested to please ask questions. If you have any question regarding the talk of this Honorable Dr. Jitrak sir, then you can ask question.
Yes, please. Any questions are welcome, because I think only 6 participants are participating in this session.
Sir, we are telecasting live on the YouTube.
Okay.
There are many participants.
There are other
They are
participants
watching live on the YouTube.
Okay. That's great.
They are, if you have, they write the questions in the comment box.
Okay. So if no questions, shall we conclude this session?
Just 1 minute, sir. We will check.
Yeah.
Thank you, sir. No, I think no questions are there.
Okay. Thank you very much.
Please stop sharing your slide.
Yeah. Thank you, sir.
Thank you, sir, for your enlightening and insightful talk on total synthesis on canodine and alkaloids. Thank you very much. Now we will proceed to next session. So I would like to request Professor P.M. Kadam sir to introduce the first person of this session, Honorable Dr. Nath More sir.
Thank you, sir. So good afternoon all. This is the task of introducing our next speaker. It is a great privilege to me to introduce such a great personality. He is a man of virtue and simplicity. So welcome our speaker, Honorable Dr. Nagnath More sir. So Nagnath More sir is working as the Marie Curie postdoctoral researcher at Department of Chemistry, University of Birmingham, UK. Dr. More sir completed his undergraduate and postgraduate education from Dr. Babasaheb Ambedkar Marathwada University, Aurangabad. He qualified a NET exam with All India rank at 24. So he also qualified the entrance test for ISER, and he completed his PhD work under the guidance of Professor M. Jagmohan in ISER, Pune. During his PhD work, he taught the analytical chemistry to the UG and the PG students. He guided them for the research work for the PG project. After PG, he worked as a postdoctoral fellow at Ben Gurion University of Negev, Israel for 2 years. During this work, he also worked on a joint industrial project with ADM, Agrochemical Industry Israel, to develop some pesticides. He has received a travel award for 2 times from European Cooperation in Science and Technology to attend the Chaos meeting at University of Athens and Cyprus. Dr. More sir, has published a number of papers in national and international reputed journals like Chemical Communication, Organic Letter, chemical journals, that is the chemical European journals, and the Journal of Organic Chemistry, and many more reputed journals. He has attended many conferences and presented his oral presentation and poster presentation in national and international conferences. He has reviewed the manuscripts of journals like Chemical Communication, Journal of Organic Chemistry, and Organic Letter, and many more reputed journals. So, friends, today we have an opportunity to listen such an young and talented personality. He is going to share his research experience on architecture of CH bond functionalization for the development of complex molecules and pharmaceutical targets. So I heartily welcome Dr. Nagnath More sir to this webinar.
Thank you, sir.
Thank you, sir. Now I would like to request Honorable Dr. Nagnath More sir to deliver his talk in this session.
I hope everyone audible for me. And Kadam sir, thank you for your introduction for me. So today we are here we are here for the one-day international webinar on new horizons in chemical research and product development, arranged by Chemistry Department of Shri Vyankatesh Arts, Commerce and Science College with the IQAC. And I'm going to talk about the architecture of CH1 interaction for the development of complex molecules and pharmaceutical targets. So we see in the couple of years, the chemistry research is changing every year, and it's taking new heights in the chemistry research field. In the last 50 years, there are many reactions, several reactions, plenty of reactions discovered in the chemical research, and they reported in the international publications. And those still we are using those chemical reactions in the discovery of pharmaceutical products and biological active chemical reactions. Now people started to editing or protein modification, and which is very important in the drug discovery. And the next 15 years, the people initiated machine molecular editing, that is machine learning, and machine tells you that by using available data where you need to edit the molecule at which point to get a biological active compounds. And this is all these fields are there where past, present and future are well balanced to advances the methods to discover pharmaceutical targets. So if we think about it, if you discuss about a discovery of medicinal chemistry, any medicine, if you see the journey of the one medicine from the synthesis to the market, if one medicine, it takes long time to come to the market, and if you see the base of this building is the synthesis. The journey of the drug starts from synthesis. So synthesis is a very important, and this is the base of this building, and that's why always synthesis is the non-divisible part of the drug discovery. So I am going to discuss some methods which are directly used to produce drugs or biological active compounds and even natural products. So people initially started the carbon-carbon bond formation reaction by using organometallic reagents with the organohalides, and these reactions have been used widely in the chemical industries as well as pharmaceutical industries. And this great success and significance of transition metal catalyst coupling reaction were highlighted by Nobel Prize in Chemistry in 2010. But even people see the drawbacks in these reactions, although they have been used widely in the academic as well as industrial chemical research. So all these methods needs highly designed, specially designed ligands and metal catalysts, and these metal catalysts are very toxic to different extent, and removal of these methods quite costly and challenging. So with these drawbacks, having these drawbacks in hand, the people started to think about something different and something exciting results in the CH bond activation. And then next, the synthetic scientists, they have started to look around the CH bond activation. The CH bond can be activated using metal catalyzed transformations, organic, inorganic reagents, photochemical methods, and electrochemical methods. This bond, CH bond, the CH inner bond can be activated and functionalized with the various functional groups. The CH bond activation become very attractive and important in organic synthesis because it's highly atom economical, cost effective, and in most cases they don't require any pre-functionalization of the starting material. So there are two types of CH bond activation. The first In 1967, Mortani and Fujiwara, they started and studied non-chelation assisted CH bond activation. So in this, the simple, if you take the aromatic compounds and they functionize with alkene in the presence of palladium catalyst. But what is the most drawback of the most and the biggest drawback of this methodology is it is not selective. It is it is atom economical and environmentally friendly, but it's not selective. You see, there are three hydrogens which the alkylation will go over all these sites. To overcome this problem, Fujiwara reported is chelation assisted CH bond activation. So what happens in this chelation assisted CH bond activation? The starting material introduces the directing groups which coordinate with the metal and directs to functionalize the adjacent of this site. And this CH bond activation known as chelation assisted CH bond activation. So this chelation CH bond activation is highly selective and atom economical and environmentally friendly. So if you see there, the direction group may be with nitrogen, oxygen, and many others. They direct the ortho CH bond to get it activate and functionalize. The mechanism of this non-chelation assisted CH bond activation, just the oxidative addition of the starting material to the palladium catalyst. The palladium is electrophilic in nature. It just undergoes electrophilic substitution with the aromatic and then next insertion of this alkene to get the final product. And the mechanism for chelation assisted CH bond activation is the introduction of functional group, and these functional groups interact with the d orbital of the transition metal and direct the ortho CH bond of the aromatic compounds, and then it undergoes the insertion of the alkene to get the alkylated product. So with this help of CH bond activation, the CH bond can be, inert CH bond can be activated and functionalized to get a variety of natural products, vital organic complexes, biological active compounds, and even can be done, protein modification can be done with the help of CH bond activation. And also, CH bonds are found in nearly all organic compounds. All we know that the CH bonds are present in simple hydrocarbons, like if you take methane or cyclohexane, there is a CH bond. And also polyurine materials like C60, you might know of C60 that contains inert CH bond and can be functionalized by using CH bond activation. Along with this, there are alkaloids which also have the many CH bonds present in the alkaloids. It can be again functionalized to modify the quinine or alkaloid molecule. And also CH bonds are present in biopolymers as well as in synthetic polymers. So this CH, it would be very interesting if we activate these CH bonds, we can functionalize these inert hydrocarbons to get a functionalized material, and even they can be used in the material chemistry as well as pharmaceutical industry. If you see in the recent year, every year there are increasing number of publications have been published in the CH bond activation. Every year the people are coming up with new ideas and publishing in the international journals. So I would like to discuss first the example of the CH bond activation, where it has been used to synthesize the direct natural products. In 2015, Itami and Davis group they studied the sequential CH bond activation to prepare dictodendrins A and F. So their direct CH bond activation, 3 steps, which are the key steps for the synthesis of these natural products. So these dictodendrins A and F. This this is the natural products which has potent inhibitory activity for beta site amyloid cleaving enzyme and have significant biological activities. How they started to synthesize these natural products? They initially started of these natural products that N-alkyl pyrrole. So first, when they studied the functionalities of this 2CH bond of this 2CH bond of this pyrrole with the diazo compounds, they got the CH functionalization at both sides, CH functionalization in the presence of rhodium. But when they tried to introduce another aryl moiety at third position of the nitrogen, third position of the pyrrole, they failed to introduce aryl moiety in the next step. So it's not easy. It's not easy. It's easy maybe for to present here, but they might took us a long time to come up with this idea. That they came with this idea that first they tried to introduce the aryl moiety, aryl group at the third position in the presence of rhodium catalyst, and then they functionalized these two CH, the remaining two CH bond with that diazo compound in the presence of rhodium catalyst, and finally with the 2-3 steps they could achieve the synthesis of dictodendrins A and F. After this synthesis, there is another functional group directed CH bond activation where they they have used or implemented this synthetic methodology to synthesize vildagliptin, which is another antibiotic drugs, and for they have tried to synthesize the analog of this antibiotic drug by using the functional group directed CH1 activation. So this simple alcohol, because the cyclic alcohol, they converted into, they protected with the directing group, and this acts as a directing to activate this adjacent CH1 in the presence of palladium catalyst, and then after having these cyclic compounds, just reduction on the with the aluminum hydride, they ended with this amino alcohol, and on further steps, 2-3 steps, they prepared the vildagliptin analog, which acts as an antibiotic. So sometimes we think that we need to prepare analog. So sometimes analog the analog of the biological active compounds may even show better efficiency in the activity. So these methods, if you see just only low loading of catalyst, that is only 1 mole percent of the palladium catalyst has been used, and the CH activation is a key steps for the synthesis of analog. So another recent example of the site selective CH1 activation using functional group directed CH1 activation. So in recently Krishna, the lab, the group, they have been studied the site selective CH1 activation. So if you take this complex starting material and you see there are many CH bonds which are inert, and this catalyst, the catalyst I have shown here, this manganese catalyst, just selects the CH bond at the carbon 3 at the shown here carbon number 3. They activated in the presence of the manganese and get selectively activation of this CH bond and to get cyclized with this functional group to get this cyclized end product. And also this methodology have been implemented to make to report a gram scale of this starting material, and they observed quite good yield, that is 75% of yield of this product. There are many natural products and biological active compounds which have been reported using CH1 activation. So all of them here, some of them I have just mentioned, and these all these are directly prepared by using direct CH1 activation, or in the synthesis of all these materials, the key steps were was used, that is CH1 activation. The one more recent site selective CH1 activation in 2020, recently the U group from the USA, they have reported site selective CH1 activation and they functionalize with aromatic halides. So if you see this purine, there are around 7 to 8 hydrogen carbon hydrogen bonds which are ready to activate and functionalize. In the literature, there are many methods to activate carbon hydrogen number 2, 3, 4, 5, even 8. But 6 and 7, as I mentioned here, number 6 and 7, still they couldn't reach to that carbon hydrogen bond to get it activated. So U group, they studied, they extended the functional group, they increased the length of the functional group, and they could reach to the selective carbon hydrogen bond to get it activated. How they did it? If they designed this kind of template, they designed this kind of template which can reach to the desired carbon hydrogen bond to get it activated. And quinoline and isoquinolines are present, these moieties are present in several natural products and biological active compounds. So it's very important to activate their selective positions, and then finally with this model, with this modified catalyst, they could achieve the activation of CH bond and they functionalize with aromatic halides to get arylated isoquinolines and quinolines. Next, I am going to talk about the protein modification, which is now recent interest among the chemical research scientific community. So here in recently the Macmillan group reported site selective or micromapping of the protein. So what is the micromapping of protein? So in the literature there are several modifications which are achieved through diffusion, or it takes these are macro mapping of protein modification. So what they have done, just they initially studied the activation of diazo compounds to get carbene to next benzylic carbene. So what they have done, just the simple iridium catalyst, they have studied the diazo compound in the presence of catalyst, and they found that these diazo compounds in the presence of light with the help of catalyst generates carbene. And just they thought that this carbene may react with another CH bond in the protein. So we know that the proteins are highly complex materials. They have plenty of CH bonds and other functional groups. And they have studied this catalyst and they observed that out of the radius, there is some nanometer, 0.1 nanometer radius around this catalyst where these diazo compounds can be activated. Out of this radius, if they try to get it activated out of this radius or out of this range of the catalyst, like consider this catalyst acts as an antenna. Out of the range of this antenna, this diazo compound doesn't activate or doesn't produce any carbene or radical. That's what they thought, that it could be very interesting and possible to mapping of the protein. So they modified this catalyst with the carboxylic group and also with unsaturated alkyne group. So why they have modified this catalyst in the cell? So to increase the lipophilicity of this catalyst, they have modified with this. And this with modified catalyst, they observe that it doesn't affect the reactivity of diazo compounds, or even it doesn't affect the yield of the production of this carbene. So these groups, they interact with the functional groups of the proteins, and then acts as a catalyst, acts as an antenna. And in the presence of light, once the light falls on the catalyst, it generates radical, and this radical interacts with or inserts with the CH bond present in the protein. So this is the micro mapping they have been studied, and the other CH bonds present out of this radius, they didn't affect with this, or they didn't functionalize with the carbon. So this is very interesting and site selective modification of protein they have been studying recently. Another example of site selective functionalization of protein, that is CH alkylation of methanol. They have studied with this simple basic chemistry and very simple catalyst. They have alkylated the carbon which is attached to sulfur, and it's very highly difficult to functionalize particularly this carbon because the sulfur is highly active and nucleophilic in nature. It easily undergoes oxidation to give sulfone or sulfonates. So this selectively, by using this catalyst, the limiflavin catalyst, they have studied this site selective C alkylation of methanol. So if you see the mechanism, it looks very simple, but this was a great achievement in the synthetic CH1 activation of synthetic organic chemistry. With the presence of the light, this catalyst undergoes from the singlet to triplet, generates N-center radicals, and N-center radicals abstract the hydrogen atom from the methyl group of the methanion and generates this radical. And this radicals undergoes the addition with the alkylating reagents, and to get the final the C alkylated methanol product. So with this optimized condition and the mechanistic studies, they extended the scope of this reaction with the variety of acceptors, variety of alkylating reagents, and in all cases they ended or they have very good, quite moderate good yields. So why this protein modification and micromapping is important in the synthetic organic chemistry? So significance of this work or this technology, that engineering of the proteins may find immediate using antibody targets and identification. That currently we are facing the whole world facing the problem of COVID-19, that where we are unable to identify the antibody target, because it's the mutation of the COVID. This coronavirus. So once you know that what is the structure of or what is the antigen of the virus, we can identify easily, and antibody can interact with that antigen and can destroy the virus. So this technology may useful in the coming years to identify the molecular target of antibody. And then this technology also can be used in signal transduction pathways. What is the signal transduction pathways? That is the health of the or status of the cell can be sent by the using signals, and these signals, just as I have shown here in the picture, the signals can be sent that the status, that whether the cell is infected or not, or just to send the message to the status of the cell. And also we can know the junction between two different cells by using this technology. And also this technology may be useful in the coming years to know the environment of many diseases. After that, just the CH bond functionalization also presented or they studied in the preparation of macrocycles. So there are plenty of macrocycles present in the market, and also they are actively working in the medicinal chemistry. So there are Here some of them I presented, these microcycles, which are people trying to study and to prepare in the efficient manner. So why this is important to report or study the preparation of or synthesis of macrocycles, the macrocycles. So bioactive molecules that is derived from, some of them are derived from natural products. So over 40 cyclic peptides drugs are in clinical use. So present, if you go into the market, drug market, you get or you see that 40 cyclic peptides are currently active in clinical use. And every year, one new cyclic peptide drug enters in the market on average. So it is very important again to make these kind of macrocycles, peptide macrocycles. So sometimes we think that why we need the modification of the present drug or the drug, whatever drug present in the market, why we need modification. So aralomycin is the drug which acts as an antibacterial drug in the medicinal use, and these drugs was already present, and then genetic the this is the pharmaceutical company wanted to study this wanted to modify this drug, modified these macrocycles to get new macrocycle. What what happened after modification of these drugs, presented drugs, aralomycin? They found that it is 500-fold more potent than aralomycin. The RLMycin, whatever the activity, biological activity was with the RLMycin, just with the functional modification of these uh, macrocycles, they found that this is more 500-fold more potent than RLMycin. So, so we can think that modification how it works and give the more potent drugs for us for in the medicinal chemistry. So there are many challenges in the synthesis of these microcycles that these challenges either need to couple these two aromatic moieties and then the connection of the amino acid group, or first try to connect this amino ends group and then try to couple these two CH phenolic moieties. So in recently, the Baran group, they studied this preparation of these microcycles. They just prepared first peptide bond, and in the presence of copper catalyst in the oxidative CH1 translation manner, they connected these two CH bonds to make these cyclic peptides. But here, what is the drawback of this method? They have been used a stoichiometric amount of this metal catalyst to overcome this problem. To overcome this problem, recently the PAPO group reported, they have been reported the successful transformation of the cyclic peptides, and then even tried to prepare the aralomycin analog of this aralomycin. They have used only 1 mole percent of iron TPP catalyst in the presence of MCPP as an external oxidant. They just first tried to they couple to the phenolic moiety of the amino acids via oxidative CH1 activation, and then they couple this amino acid to get a cyclic peptide. So there are many challenges in this synthetic chemistry that still we need to explore: site selective modification of biomolecules, synthesis of nanocanonical nucleosides, and direct CH coupling of electron deficient phenols. That nitrophenols even not reported direct coupling of nitrophenol without using pre-activation. And I am sure that if academic research with their fundamental knowledge, fundamental basic research, if you collaborate with the academic research, collaborate or join with industrial research with enabling technology, we can it can be the much more will come out to benefit science and society to solve the solve major issues in the world. And this can be there would be output that development of efficient and sustainable methods for the product development by joining the two research community, that is academic and industry, and also even can be produced and even more biological potent drugs and structurally simple drug molecules, and all together we work can work to make better and better life on the earth, maintaining healthy environment. And I would like to thank to organizing committee of the Sri Venkatesh Arts, Commerce and Science College for this invitation, and all members of chemistry department of Sri Venkatesh Arts, Commerce and Science College. Thank you.
Thank you, sir, for your informative and insightful talk on architecture of CH bond functionalization for the development of complex molecules and pharmaceutical targets. Thank you, sir. Is there any question from participants? Sir, participants are asking questions in comment box. So,
Okay.
there are very few participants are there on Zoom. So participants are asking on YouTube. Okay. If participants have any question, then they can ask question. So I think there is no question regarding this talk. So thank you, More sir. Sir, please stop sharing the screen. Please stop sharing the screen. Okay. Thank you, sir. So now we will proceed to next session of this webinar. So I would like to request Professor P.M. Kadam sir to introduce the resource person of this next session, Honorable Dr. Andrew Virnapur sir.
Thank you, sir. Good afternoon. So it's my great privilege to introduce our next speaker. So I feel elated to introduce him to everyone today. After all, he is a living inspiration to the young ones and the young ones. So I welcome our next guest, Dr. Verla Andrew Virnokar. Sir has a teaching experience of more than 18 years. He completed his PhD in analytical environmental chemistry in 2012. He has published more than 60 research papers in various national and international journals. He has published 9 books. He has published chapters in various books. He has presented papers in various national and international conferences. He also has acquired or delivered a lecture as a resource person in various national and international conferences. He is a founder coordinator of a group research in analytical chemistry, environment and climate change. He is a life member and fellow Indian Chemical Society, associate member of a Royal Society of Chemistry, and the member of various scientific society. So the list is very large. So I, due to the time, I can't read all the names here. So sir is the members of various academics. He is an editor of various journals like Science View Journal, Research Journal of Chemical Science, Einstein International Journal of Applied Science, International Journal of Forensic Chemistry, Lexicon, and International Journal of Science, and many more journals. So he has written various technical reports like environmental impact assessment of a pattern farm expansion project at Useria community, Edo State, and many more technical report has written. He is also involved in the various social activities. He has worked as an external examiner for the PhD examination. He has got a various research project grants funded from the various governments. So I welcome such multi-talented personality to Dr. Verla Andrew Virno. Sir, please start your video and audio.
Thank you, sir.
Thank you, sir.
Now I
Now
would
I would
like
like
to
to
request
request
Honorable
Honorable
Dr.
Dr.
Varla Andrew
Virla
Virnakar, sir,
Andrew Virnakar,
to
sir,
deliver his
to
talk
deliver his
in this
talk
session.
in this session.
Sir, you can share your presentation.
अरे वो व्हाट्सएप में मैंने जो दूसरी इमेज भेजी है ना वो लो, पहली वाली मत लो।
Sir, can you hear me? Verla sir. We are trying to connect our guest, so just wait for a few minutes. There is some technical issue from his side. So just take a few minutes. We will try to connect again.
Use Alexa.
Listener is trying to connect, but there is some technical problem. So wait for a few minutes.
Dear participants, we have shared the feedback link form on the YouTube. So please fill the feedback form. It is active till 5 p.m. today. So I think there is a connectivity problem. So we will now forward to conclude this webinar. So I request Kale sir to continue the webinar.
Now, Honorable Dr. Chavan to tell the president of this.
Hello.
Yes, sir.
Hello. Good afternoon, everybody. At the outset, I wish everybody a very happy new year. I wish everybody that the coming 2021 would be very happy and prosperous for everyone who have joined this webinar. The source of inspiration, Honorable Chairman of the institution, Raja Vijay Singh Jadhav, the greatest personalities and the eminent persons who joined this seminar as the resource person, Dr. Dattatray Deulgaon sir from USA, Dr. Nagorao Dhatrak sir from Pune, Dr. Verla Vinko from Nigeria, and Dr. Nagarath Vaimore from UK. The one-day international webinar organized by the Department of Chemistry in Shri Vyankatesh Arts and Commerce College, Deulgaon, has been a very successful event just because these three eminent personalities expressed their valuable opinions on the topic given to them. The title of this webinar was New Horizon in Chemical Research and Product Development. The Department of Chemistry in our college has been very much active in research activities. And so certain things should be always discussed, or some new vista or new horizons should be opened to the researchers, not only in the college, but to the researchers all over the world, has been the motto of the Department of Chemistry in our college. Hence, this webinar is organized today on the same topic. The trio, Dr. Deulgaon sir, Dr. Dhatrak sir, and Dr. More sir, have really contributed much with regard to the topics which had been allotted to them for discussion. In the present scenario, it is really very essential that such topics must be discussed in the webinar, because day by day, the research in our country as well as all over the world has been set back just because of the interest of the people. But if such people are really interested in such activities, I don't think that the research would be stopped or research would be discontinued. Rather, the research would go fast. Because when I listen to the lectures, when I listen to the address of these three eminent personalities, I really realize that there is a lot of devotion of such people in the research of chemistry and some other science subject also. But the research in chemistry, particularly with respect to this webinar, is really helpful. And in all three deliberations, we came to know that how research and research in different objects or in different product is essential in the present scenario has been convinced to us or has been told to us by these three greatest personalities. We are really blessed by having such eminent guests abroad and across the sea as a chief guest and the resource person of our webinar. It's a matter of great pride to all of us that really they accepted our invitation and spared their time. Though the time in India is different from the time in their own country, they consented for this webinar, and they not only consented but also expressed their valuable opinions with respect to this topic, with respect to this webinar. It's really a matter of great pride to all of us, and we feel pride and we feel great pleasure to let all the people, all the participants know that we could contribute some views with the help of this trio, with the help of this three-man person on the topic that is new horizon in chemical research and product development to the people who are interested in research activity, particularly in chemistry subject. I hope that this webinar would definitely help all the people who have joined this webinar today, because research is the acute need of the time, and such deliberations would definitely proceed with the help of such kinds of discussions and deliberations that took place today with the guidance of Deulgaon sir, Dhatrak sir, and More sir. I congratulate the Department of Chemistry in our college for organizing such nice event, and I express my deep sense of gratitude to all resource persons, all eminent persons or eminent scholars abroad who really made this webinar a very successful one. With this remark, I conclude and I stop here. I thank to the Department of Chemistry for giving me an opportunity to preside over this webinar. And with this remark, I stop here. Thank you. Thank you very much.
Thank you, sir. Thank you. Now I request Dr. D.R. Birade to propose vote of thanks of this international webinar.
Good afternoon to all most valued guests, delegates, work teachers, and all the participants of this one-day international webinar titled New Horizons in Chemical Research and Product Development, organized by the Department of Chemistry in collaboration with IQAC, Shri Vyankatesh Arts, Commerce and Science College, Deulgaon, Raja. I would like to take this opportunity to express my gratitude and propose vote of thanks on this occasion. As a co-coordinator of this international webinar, on behalf of our college, first of all, my sincere thanks to chief guests and resource persons who spared time from their busy schedule to grace this occasion. I would like to thank to Dr. Dattatraya Deulgaon sir, Scientist I, Department of Radiology, the University of Alabama, Birmingham, USA. Thank you, sir, to share your experiences and illuminating speech. I would like to propose hearty vote of thanks to Dr. N.R. Dhatrak, sir, Department of Chemistry, Savitribai Phule Pune University, Pune. Thank you, sir, for sharing valuable information and research address. I extend my most sincere thanks to Dr. Nagnath Vaimore, Marie Curie postdoctoral researchers, University of Birmingham, UK. Thanks, sir, for your discourse. I also thank to Dr. Verle Andrew, sir. We are unable to listen them, but again, I would thank to him. Further, a big thank you to the inaugurator of this webinar, Sudhir sir, IQAC coordinator, head of the department, and I would like to propose hearty vote of thanks to all the delegates and students who have joined from different parts of the world to this one-day international webinar. Last but not least, I would like to express my sincere thanks to organizing committee, head, Department of Chemistry, and all the faculty members for this help and support. With due permission of Honorable Principal, sir, I hereby declare that this webinar is over. Thank you.
Thank you, sir. We have shared the feedback link on YouTube. So participants are requested to please fill this feedback form till 5 p.m. today. Okay, thank you. All the participants will obtain their certificate.
All the participants are requested to submit the feedback form. The feedback forms are active till 5 p.m. today, and the certificates will be issued or sent on the Telegram group. So the participants who are not joining the Telegram group, please join the Telegram group. The certificates will be issued or sent in only the Telegram group. So we are not individually sending the certificates on the emails. The certificates will be sent only in the Telegram group. So the participants are requested to join the Telegram group